I'll be taking you through the small RNA analysis features in Avadis NGS. So the Avadis NGS product is built for use by biologists. It's built on the same user-friendly platform as GeneSpring. So if any of you are GeneSpring customers, the layout of the tool will be very familiar to you. Even if you're new to the platform, it is a very intuitive interface and you should be able to get started with very little effort. Avadis NGS import of aligned files in multiple formats. You can import SAM files, BAM files, and you can also import data in aligned format which is specific to the Illumina platform. After you import this aligned data into the tool, depending on the type of experiment that you start off, you'll be presented with a customized workflow for ChIP-seq analysis, RNA-seq analysis, DNA-seq analysis, or small RNA analysis. And this time, in the upcoming version of Avadis NGS, we are also including an aligner which is capable of aligning small RNA data and DNA data. In today's webinar, some of the questions that will be addressed are: how is small RNA data imported into the tool? What are small RNA specific QC and filtering steps? How is small RNA expression quantified? How are the expression values visualized? How are miRNA targets identified? How are novel small RNA genes discovered and classified? And how do you find annotation discrepancies since it's still an emerging field? So let me start off with how is small RNA data imported into the tool. Avadis NGS supports import of aligned files. So if you have data that has been aligned by any aligner and it's present in SAM format, BAM format, or in Illumina specific aligned formats, you can import the data and get started with small RNA analysis. In addition, if you have raw files, that is if you have FASTA, FASTA-Q or counts files, you can import those files into the Avadis NGS aligner, and from the alignment results you can create a small RNA analysis experiment. So let me get into the details of why we decided to include an aligner this time in the tool. For that, let's take a look at how our small RNA reads generated typically. Usually we start off with a total RNA sample. We use size selection and select molecules which are maybe of 15 to 30 nucleotides long. After that, we use a 3' RNA adapter and we ligate it towards the end of these molecules. Then we carry out sequencing for a fixed number of cycles, let's say 35 cycles. So then we have reads where the first part of the read represents bases from the RNA molecules and the later part of the read contains data from the adapter sequence. In this particular example, I have shown the bases coming from the RNA molecules using blue letters and the bases coming from the adapter in red letters. Now once we take this data for alignment, the aligner does not know what part of the read came from the original RNA molecule and what part of the read came from the adapter. But if you provide it with the adapter sequence, then we can detect presence of the adapters in the latter parts of the reads. In this particular case, those parts have been highlighted with red, and you'll notice that because errors are possible even in the latter part of the read, we need to be able to find the adapter sequence even if there are sequencing errors. After that, the adapter needs to be trimmed out and we are left with variable length reads which then need to be aligned against the genome. So because of these multiple steps, in other words, finding the adapter, trimming the adapter, and being able to align variable length reads, we decided to include alignment as a new experiment type in Avadis NGS for the upcoming release. So let's take a look at what the alignment experiment looks like. We create a new experiment. From the experiment type drop down, we choose the alignment small RNA type. After that, we choose the organism and build against which the reads need to be aligned. And then we select the files containing the reads that need to be aligned against the genome. The drop-down shows multiple formats that are supported by the aligner. You have the familiar FASTA and FASTA-Q files. In addition, we also have text files to support the counts data, which is used often for small RNA data representation. So the counts data looks somewhat like this. It is obtained by taking a FASTA or FASTQ file, ignoring the qualities, simply taking the read sequence and counting how many occurrences of that unique read sequence are present in the file. For instance, here the read sequence comprising entirely of A's is present 66 times in the first file. It is present 467 times in the second file. So the counts file format is a very compact way of representing the distinct sequences present in the reads file, but it ignores the qualities. Once you import files in these particular formats into the aligner, it then shows you an alignment experiment which has a workflow on the right hand side. The workflow has a pre-alignment QC section where you can inspect the base quality distributions and the read quality distributions. Based on these pre-alignment QC metrics, you can then choose parameters with which to run the alignment, and after alignment is carried out, you can carry out the post-alignment QCs. Once you click on Run Alignment, you'll be presented with a dialog where you can place the alignment parameters. So you can choose the number of mismatches which need to be tolerated, the number of matches you want to report in case of multiply matching reads, etc. After that, if it is a small RNA experiment, you will be shown a dialog which is specific to small RNA alignment. So here you can give the 3' RNA adapter which needs to be trimmed out. A list of common adapters will be shown in the drop-down. If you have an adapter which does not match any of the ones shown in the list, you can type in your own adapter sequence. You can also specify the number of mismatches that need to be allowed in the 3' adapter section. After that, the alignment is carried out and the navigator shows the result of the alignment process. You can inspect the reads in the Genome Browser, you can do post-alignment QC, and if you want, you can rerun alignment any number of times. And once you're happy with the results of alignment, you can directly create a small RNA analysis experiment from the results of the alignment. So let us see what a small RNA analysis experiment creation looks like. You choose the experiment type as usual. After that, you provide the annotations and metadata using which analysis needs to be carried out. So you specify the organism in the build and the small RNA annotations, which are pre-packaged annotations available from our annotation server. In addition, you also need to choose a transcript annotation. This is because if you carry out target prediction analysis, the result is a list of protein coding genes, and as you're aware from our RNA-seq experiment descriptions, we provide annotations for UCAC genes, RefSeq genes, and ensemble genes. So here you're choosing the gene model against which you want to carry out the target prediction analysis. After filling in this data, you're then shown a dialog box where you can choose the samples that you wish to analyze. In this particular case, the samples had been aligned with a different aligner. They were in SAM format and we're importing them into the tool. If you are starting from an alignment experiment, these steps would be skipped. You also have a choice of renaming your samples. And after that, the samples are imported into the tool. You are shown with a navigator containing the imported samples, and the Genome Browser shows one track for each of the samples that you imported. Let's now take a look at the small RNA analysis workflow. The first part of the workflow is where you set up your experiment grouping and interpretations. You can carry out a quality inspection. Based on the results of the quality inspection, you can filter out reads of poor quality. After that, typically you'll carry out quantification. And once quantification is carried out, using your interpretation, you can do differential expression analysis to find out lists of small RNA genes which are behaving differently between different conditions. The differential analysis step is exactly the same as RNA-seq, so I will not be spending much time on it in this particular webinar. In this particular webinar, we'll be concentrating only on features which are unique to small RNA analysis. After we find a set of differentially expressed small RNA genes, usually we'll use find targeted genes to find the protein coding genes which are the targets of these. And after that, we can carry out the familiar Go analysis, GSEA, pathway analysis, etc. So let's take a look at the QC and filtering steps which are unique to small RNA analysis. In the other experiments, the quality of the alignments is shown as an alignment score plot, where the alignment score is a number between 0 and 100, which captures the number of mismatches and insertions and deletions in the read sequence. And alignment score is an appropriate metric when the read sequence is long, but because small RNA reads are only between 15 and 22 bases long, we provide a more intuitive mismatch count distribution. So here you can see that there are 2.75 million reads with 0 mismatches, approximately 600,000 reads with 1 mismatch, etc. Another QC metric we provide is based on annotations. If you recall, there were two different annotations that were selected at the time of experiment creation. One was the small RNA annotation and one was the protein coding gene annotation. Using those two different annotations, here we depict the number of reads falling in the small RNAs and the different classes of small RNAs. Here you can see that 37.2% of the reads are falling in annotated miRNA regions. There's a smaller fraction of reads falling in scRNAs, SNORNAs and other regions. One slice to watch out for is the exonic section. That is usually indication of degraded mRNA molecules. A large number of intronic reads could indicate the presence of novel small RNAs which might be present in the experiment. Similar to this QC step, there is a corresponding filter step, and in the filter step usually we filter out all the reads falling in exonic regions. After we carry out these filters and any of the other QC filters which are present in all the other experiment types, the read list is now ready for quantification. So in quantification, let us try to answer two questions. What do we want to quantify and how do we want to quantify it? In the small RNA annotations, the annotations indicate the precursor gene on the genome. In the case of precursor miRNA genes, the resulting transcript folds into a hairpin structure. Once the dicer enzyme cuts away the loop, one strand of the duplex participates in mRNA silencing as part of the RISC complex. In this particular case, the duplex structure consisted of the red and the blue stretches of the hairpin, and the blue stretch has been shown as participating in the targeting of a mRNA molecule. So clearly we would like to quantify how many reads are arising from the blue section which will participate in the targeting of the mRNA molecule shown out there. So we would like to quantify the reads originating from the blue portion of the precursor gene. Similarly, we'd like to quantify the reads originating from the red portion because that is also a candidate for participating in the silencing process. So these regions are what I'll be referring to as active regions of the genome in the rest of the presentation. In addition, we'd also like to quantify the total number of reads falling in the entire precursor gene, because for other classes of small RNAs such as SNORNAs and scRNAs, there are no fixed active regions, so we want to quantify the total reads coming from the entire gene. So to summarize, we want to quantify the total reads falling in the entire precursor gene stretch. In addition, we want to quantify the reads originating from certain active regions of the gene. So let's see how do we want to quantify the genes. Here I've shown a precursor gene in blue, and I've shown different reads, 4 different read sequences that align against this section of the genome. There are 100 reads which overlap partially with the gene. There are 200 reads which fall entirely inside it. There are additional 300 reads which fall entirely within the gene's boundary, and then you have another 50 reads which almost fall within the gene's boundary. So one way to quantify the expression of this particular gene would be to limit analysis only to reads which fall entirely within the gene's boundary, which would be 200 plus 300, which is 500. But because annotations are not very exact, we would like to give a little bit of leeway in this quantification process. So we allow the user to specify a delta, and we take all the reads which fall within a delta neighborhood. In a sense, we pad the gene's boundary by a delta amount, and after that padding is carried out, any reads that fall entirely within the gene's boundary count towards the quantification of the gene. In this case, now the gene's count would be 200 plus 300 plus 50, 550. We also need to take care of multiply matching reads. If the 300 reads are also participating in matches against a different gene, then the read contributes only 1/2 to each location. So after that, the quantification of the left-hand side gene would be 200 plus 150 plus 150, which is 400. So quantification needs to take into account padding, which is user customizable. It also needs to handle multiply matching reads. So now that we have seen how to quantify genes, let us see how to quantify active regions. Here I have shown a precursor gene with a single active region in it. In other words, the active region will give rise to a mature RNA which will bind against a particular target site. Now in the binding of the mature RNA to the target site, the bases from 2 to 8 bases of the mature RNA are very crucial to the binding process. So if we had a lot of different reads originating from the gene, and I've shown 5 different such read sequences, the read sequence at the bottom exactly matches the active region boundaries. So its 5' end matches the 5' end of the active region, the 3' end matches the 3' end of the active region. The second read and the third read, they match at the 5' end, but they do not match at the 3' end. And the first 2 reads at the top do not match the 5' end also. Now because we know that in binding to the target site, the bases from 2 to 8 are crucial, it is reasonable to take the counts of the bottom 3 reads, since they have exactly the same 2 through 8 bases, and use them to quantify the active region's value. So if these were the different read counts, then the default quantification in Avadis NGS would assign a value of 200 plus 100 plus 70 to this particular active region. However, you do have an option of restricting analysis to only the exact matching reads. So you could say that only reads which exactly match the 5' and 3' boundaries of the active region should contribute towards the active region's count, in which case you'll get a value of 200. So let's see how these dialogues look in Avadis NGS. We select the reads that need to be quantified. You have an option of carrying out novel small RNA detection during the quantification process, or you could choose to stick with only those small RNA genes which are present in the annotations that you downloaded. And the second option is where you specify whether you want to be very conservative in quantifying active regions. That is, you want to restrict analysis to only those reads which exactly match the active region boundaries, or whether you want to use slightly more liberal values that I discussed earlier. So based on those parameters, counts are going to be assigned to each of the reads, but then you can choose one of several normalization techniques. We support DE-seq, TMM, quantile, and a simple sample count based normalization. After you choose these parameters, quantification is carried out and the navigator shows the entity list. There are multiple entity lists shown in the navigator. The all genes is the entity list of all of the precursor genes, essentially the entire stretch. The all active regions comprises of only the active regions of mRNA genes. The all genes further has 2 children below it. One is the known genes, that is the genes that were present in the annotations, and the novel genes that were detected during quantification process. So now that all of the elements have been quantified, let us see how to view the quantification results. The simplest result is a spreadsheet where for every single gene, for each one of the samples, we have the normalized expression value of that particular gene. So here I have highlighted HSA-MIR130B, and we have 14 samples, but only around 12 of them are shown in this particular view, and the value of the gene in each one of the samples is shown in a single row. You can sort on the spreadsheet, you can sort on any particular sample, and you can click on that particular gene of any gene of interest to see more details. One thing you would like to do when looking at this particular spreadsheet is see the profile of the gene across the multiple samples. Here there are 14 different samples. It's hard to just look at the spreadsheet and get a sense of in which samples is it up and in which samples is it down. So there's a profile plot which shows for the selected gene the profile in the different samples. After that, if you want to look at the actual reads that are contributing towards the gene's expression, you could try to look at it in the Genome Browser. Here I've shown only one of the read tracks. If you recall, there were 14 different samples. This is the Genome Browser track corresponding to a single sample, the 2-day sample. The track at the bottom shows the small RNA structure in dot parenthesis notation. It also has it marked in the darker blue bands, the 2 active regions. Those are the ones which will be pairing in the hairpin structure. And the reads falling in this particular genomic region are shown in the Genome Browser. But as you'll notice, the coverage is very high. It's 4,300. So you can't really see all of the reads without scrolling up and down. So you don't get a sense of how many reads are exactly corresponding to the active region, how many of them are matching the 5' end of the active region. It is hard to get a sense of all of those from the Genome Browser. Because of that, we have a special small RNA gene view. Here, the left-hand side shows all the distinct read sequences. It also shows in blue and red the locations of the active regions. The sequence of the genome and the dot parenthesis notation are there as column headers. And on the right-hand side, you can see the count of the read in each one of the samples. So if we take a closer look, the very first read sequence starting with CAG-TGC and ending with CAT, there are 2,639 occurrences of that particular read sequence in the first sample. There are 1,272 occurrences of that read sequence in the second sample, etc. So this particular view shows you the read sequence of all the reads falling in that particular genomic region. In addition, for each read sequence, you can see how many such reads were present in each of the samples. You can click on any row in this table, and each row corresponds to a distinct read sequence, and immediately it shows the location of that read sequence on the backbone structure of the small RNA gene. So small RNA structure is shown at the top. By default, it has the active regions marked out in red and blue, but any row that you click on out here, it is lassoed onto the small RNA gene view. The structure is also zoomable. If you notice, it's at 54% zoom out here. With a scroll mouse, you can zoom in and out of that structure and examine it in greater detail. This view is in a sense similar to the variant support view that we have in SNP calling. There also all the identical read sequences were compressed into a single cluster. Here we are attempting to do something very similar. If for a particular read sequence you want to investigate its profile, essentially you want to take the numbers on the right hand side spreadsheet and plot them as a profile plot. That is also shown in the small RNA gene view. So to summarize, this is what the small RNA gene view looks like. There's a spreadsheet at the bottom where you can sort on any of the sample headers. You can click on any row in that spreadsheet. Details of that particular gene are shown on the top half. All the reads that are falling in that genomic stretch corresponding to the gene are shown in the panel just above the spreadsheet. The counts in the different samples are shown in the right. The read profile plot is shown at the top left corner. The plot of the genes count across the different samples is shown in the top right corner, and the structure itself is shown above it. So it's a comprehensive view providing complete details of any particular small RNA gene. So after you have done quantification and performed differential analysis, usually you have a smaller set of mature RNAs, and you want to find out what the targets of those mature RNAs are. We provide target prediction, multiple target prediction databases from our annotation server. You can download those through the annotation manager, and whichever annotations you download, those annotations are shown in this particular dialog box. So you can choose the target prediction database that you're most comfortable with, Target Scan, Pictar, or the other target prediction databases. You can also decide whether you want to only consider targets present in all of the databases or a subset of them. In a sense, you can do a union or intersection of the target prediction databases. After that, a hypergeometric test is carried out, and for each of the targets, a p-value is assigned saying how significantly enriched it is. Now each one of the target prediction databases, they also qualify their prediction with a lot of parameters, conservation score, context score, etc. So you can choose to filter each one of these databases using the parameters before carrying out the analysis. We are planning on supporting 5 target prediction databases. They are Target Scan, Pictar, Starbase, microRNA.org, and PETA. The target predictions will also be available as a track in the Genome Browser. The spreadsheet of the target prediction site is shown at the bottom in the Genome Browser, and it contains all the information present in the original site, including the context score, whether it's conserved or not, etc. All of those parameters can be used to color, filter, and browse the target prediction sites within the Genome Browser. One of the main reasons for carrying out NGS analysis is to discover novel small RNAs and genes. The novel small RNA discovery starts off by looking at sections of the genome which have a lot of reads, but there is no annotated gene in the vicinity of that enriched region. Suppose you had one such enriched region. We could call it a small RNA, but then we would have the question of what type of a small RNA is it? Is it a microRNA gene? Is it a snRNA gene, etc.? So in the novel small RNA discovery process in Avadis NGS, we first find these enriched regions, then we try to classify the enriched region as one of the specific small RNA gene types. For this, we use a block-based approach. Suppose we had a cluster of reads as shown at the bottom. We would first analyze the cluster of reads and break it into blocks. In this particular case, we would break it as 4 distinct blocks, and the blocks could overlap with each other. So each one of these blocks has a different number of reads. The very first block has a very small number of reads. The second block has a slightly high number of reads. The third block has a lot of reads, and it overlaps partially with the fourth block, which again has a small number of reads. The reason we do this detection of blocks is that different small RNA classes exhibit different block patterns. For instance, here we have 3 small RNAs, an miRNA, and a tRNA, snRNA. And if you look at the block structure, in the miRNA out here, we have one block with 132 reads, which is well separated from a second very high block with 1814 reads, which overlaps partially with a much weaker block. In the case of tRNAs, you have a block with 741 reads. It overlaps partially with a block with 17 reads, and both of these overlap with a block with 19 reads, which in turn overlaps with a very strong block again, which has 764 reads. So if each small RNA class has a different block pattern, then the pattern of these blocks can be used to classify a novel region into one of these gene types. So a paper published last year has done an analysis of the block pattern and come up with multiple features. So for a particular enriched region, after we find out the blocks, we could find out how many blocks are present in that enriched region, what is the length of that enriched region, how many of those blocks overlap with each other, what is the maximum distance between the blocks, the minimum distance, how many nucleotides are covered by multiple blocks, etc. So each of the enriched regions could be annotated with all of these block-based features. So the novel detection method proceeds as such. We start off with the known small RNA genes. For each of the known small RNA genes, we look at the reads falling within the gene's boundary and we compute the block features for that particular known small RNA gene. So based on all these known parameters, we build a decision tree based model. We run the model on all the novel regions which do not overlap with any gene, and we assign a class label and confidence to the predictions. So once this is carried out, we have novel genes in the navigator. In addition, we also have a novel detection report. The novel detection report indicates the start and end of the novel region. It indicates the predicted gene type and the confidence with which the gene type is being predicted. In addition, we use the conservation track which is there on an annotation server and find out the average conservation in this particular region. The novel detection report can be dragged into the Genome Browser. You can see the novel predicted snRNAs out here, and you can see that the conservation is very high in those two regions. In addition to finding completely new small RNAs, we would also like to verify the annotations that are present on the annotation server. The annotations that we put up for small RNAs are derived from MIR-base, Ensemble, and RefSeq, but the annotations are constantly changing, and there's no guarantee that the annotations on the sites that we obtain them from are indeed accurate. So let's see how you try to validate the mature RNA annotations using the data in your experiment. Let's go back to this picture that I showed a few slides ago. It shows a precursor miRNA gene and an active region, and we have different reads aligned against that particular stretch of the genome. We would like to find out how many reads out here do not match the 5' end of the active region, how many reads do not match the 3' end of the active region, and how many reads have mismatches in the active region stretch. All of those are indications that the annotations could be wrong or that something different is happening in this particular sample. In this particular case, we would say that the reads that do not conform to the annotations at the 5' end, there are 80 such reads. At the 3' end, there are 200 such reads. Okay. So there is a utility in the tool which carries out this analysis for every single active region, and you can choose which of the annotation discrepancies you want to report. For instance, you could say that analyze only those active regions which have at least 50 reads aligning against them, and report it only if a large fraction of the reads are discrepant. Once we get this particular report, we can click on any one of these genes, any one of these rows out here which corresponds to an active region, and we can go back to the gene view, and we can notice in the gene view whether indeed the reads are not matching the active region. For instance, out here, you'll see that the blue region demarcates the boundary of the active region, but there are only 3 reads in one sample and 4 reads in the other sample which conform to these boundaries. Whereas you have 50 reads in one sample and 19 reads in a second sample which are off by 1 base. That is, they start 1 base before the annotated region. So this could be an indication that the annotation is incorrect, or it could be an indication that something unique is happening in your particular sample. So that brings us to an end of the small RNA analysis webinar.